Hi everyone. Thank you to the organizers for the invitation for presenting some of our work. Our group focuses on the development and application of computational methods, largely from machine learning, to examine problems in gene regulation and gene regulatory networks. So I think we all know what a gene regulatory network is, but just so that we are on the same page, let me define that briefly. So gene regulatory networks determine which genes must be expressed where and when in a cell, and they are the key components of the control machinery in cells. They are composed of regulatory proteins such as transcription factors and other regulatory proteins such as signaling proteins and chromatin remodelers that regulate the expression of a gene. And we depict these relationships as directed graphs as shown as in this picture here. So in our group, we've been interested in different aspects of gene regulation and have developed a number of computational tools to look at these different problems. For example, methods to do genome scale gene regulatory network inference, methods to look at 3D genome organization, as well as how regulatory networks and regulatory programs change across long evolutionary timescales. In today's talk, I will talk about gene regulatory networks on cell fate specification, which is something that we have been increasingly getting interested in. So we know that for dynamic processes such as differentiation and cellular programming, cells transition from one state to another, and these transition states can represent different types of cell types, such as a pluripotent cell type or a committed cell type. And we know that these transitions are governed by underlying gene regulatory networks. But what we don't know is what these gene regulatory networks are and how they change across these different cell states. And so in order to really understand this problem, we need to be able to define these gene regulatory networks and look at their dynamics. And with the advent of single cell genomics, we have a lot of opportunity to answer this question in a systematic way. So let's think about the key questions that we need to answer to examine gene regulatory networks on cell lineages. So the first question is, what are the cell types or the cell states, and how are they related? And then once we have this information, what are the gene regulatory networks for each of these different cell types? How do they change? And last but not the least, how accurate are these gene regulatory networks? So let's look at the first question of what are the cell types. And now this question of defining cell types is a very popular one, and there are different approaches. A common workflow for this is as follows. We start with some single-cell RNA-seq data matrix, and we do dimensionality reduction followed by clustering, and then annotation of the clusters, which typically entails doing some additional computational / manual analysis and leverages additional resources such as gene marker lists as well as cell atlases. The challenge, however, arises when we have data from multiple samples, and these samples can come from different time points or different conditions, and we would like to be able to identify cell types from these types of conditions. And there are several approaches for trying to do dimensionality reduction and integration from multi-sample data, but the majority of them actually project the data into a single lower dimensional space. And this can be problematic when the data sets that we are integrating are very diverse. And one extreme case of that is when we are looking at across species. And in fact, there was a very recent benchmarking study, which was very comprehensive and that looked at a large number of methods and different platforms. And one of the things that they found was that integration across different species is among the challenging tasks currently. Among different dimensionality reduction methods, non-negative matrix factorization has been a powerful and popular approach. So let me tell you a little bit as this builds on some of the work that I'll describe in the subsequent slides. So in NMF, what we do is we are given an input matrix X, and we want to decompose this matrix as a product of two lower dimensional matrices U and V, which have a lower rank, and they should be able to describe the signals in our input matrix as well as possible. And another property of non-negative matrix factorization is that it handles non-negative data. So this NMF is very suitable for single-cell RNA-seq datasets, and this has been used for a number of applications. And in this setting, our input matrix is our cell by gene matrix, and the decomposition U and V tell us the low-dimensional representation of cells and genes. And then importantly, we can actually use the U and the V matrices to define cell clusters as well as their corresponding gene clusters. And oftentimes, this is a pretty good clustering of the data, although it can be refined by additional post-processing, such as graph clustering on the factors or k-means. And we have actually applied this approach to single-cell RNA-seq data, looking at early human spinal cord development. And we have identified several cell types based on expression of known markers as well as novel cell types, as you can see. And if you're interested in this work, I encourage you to read this preprint that I've listed at the bottom of the slide. Now, NMF has been used also for multi-sample data. And one simple approach to do NMF is to concatenate the data matrices. So we have genes along the columns and cells along the rows, and then apply NMF to this larger matrix. So that's called simple joint NMF or joint NMF. And another approach is used in the Liger approach, where it is assumed that the data sets that we are trying to integrate have a common shared set of factors as well as some data set specific factors. And this is basically the framework of integrative NMF. And this approach, again, is able to do a very good job of handling multiple types of data sets. However, the existing approaches typically don't handle the task or the data set heterogeneity, which can arise when we have data sets of different types of relationships, such as across species or on a time course. So we developed an approach called tree-structured matrix factorization. And in this approach, each data set, here we are showing 3 data sets, 1, 2, and 3, get their own factors, U and V. And what we do is we make the factors on the gene space to be similar to each other by putting in a regularization and make that similarity such that it changes depending upon how the data sets are related. And this information is provided as input to our framework. And at the bottom is the objective. And here alpha is the parameter that we use to try to control how much we want to impose the similarity between the different data sets. And if you're interested more in this approach in more details, please check out this poster, number 34 in the RegSys session. And for another take to this problem, I encourage you to listen to Erica Diane Lee's talk again in the RegSys program. So we applied TMF to different data sets, and we assessed the performance of TMF for clustering quality as well as for batch effect correction. And here we compared TMF to several other methods, including Liger, Scanorama, and Harmony, and some other variants of non-negative matrix factorization. And what you're seeing here are results based on clustering quality, as well as for how well the clusters actually represent real cell types. And this information was available for us for this particular data set, the PBMC data set, which is where we have 2 data sets of 2 different platforms. And what we can see is that, so these numbers here correspond to the ranks of each of the methods. And we can see that the TMF approach is actually doing quite well, ranking in the first 1 or 2 methods. Then we looked at a more complex data set where we have data from different human retinal organoids. This is complex in the sense that they come from different lines and they have different ages and different genetic perturbations. And we provided the relationship across these data sets using this hierarchy. And here again, we see that TMF is often ranking among the top methods. However, this data set also shows that there is no one method that is actually very good. And then finally, we also looked at how different methods work on a multi-species data set. And here, this data set is from a paper from Canton et al., which looked at different brain organoids from different species. And here again, we see that TMF has a pretty good performance. So we wanted to dig deeper into the question of the extent to which the different methods are able to pull out conserved clusters across different species. And so to look at that, we looked at for each of the different clusters, the extent to which the different cells from different species are actually represented. So in these white-red heat maps, what we are showing is the proportion of each of the cells from a particular species in the cluster, and we would like this to be more red across all of the different species. But this can be susceptible to the number of cells that are in the species. So we also looked at an enrichment score or a conservation score, which looks for the tendency of cells from a particular species to be enriched in a particular cluster. And based on that, what we see is that on average, the fold enrichment that we get from the TMF approach is higher than the other approaches. And you can see that methods, some of these methods are able to get much more species-specific clusters rather than getting conserved clusters. So overall, based on these evaluation metrics, we can say that TMF is able to effectively integrate data across the different heterogeneously related data sets. Then we looked at the genes that were associated with each of the different clusters, and we compared them to known marker genes of different neuronal cell types, and we were able to ID several of our cell clusters to different cell types that are present in the neuronal cell populations. And then we also tested these genes for enrichment of gene ontology processes, and we found several relevant processes that are highlighted in these red boxes. And importantly, they were also conserved across these different species. So with that, I want to conclude the first part of what I talked about. So for cell type identification, dimensionality reduction and clustering is a common first step, but it can be challenging when we are looking at heterogeneous samples. And to address that, we developed TMF, which is based on multitask matrix factorization that enables us to look at different heterogeneously related data sets. And we applied this to neuronal organoid data and found several conserved cell clusters along with different genes that were enriched in these different clusters and were also conserved across different species. Now let's look at the second question of what are the underlying gene regulatory networks for each of the different cell types, and how are they changing as we go from one cell state to another cell state. So this requires us to infer gene regulatory networks from single cell omics data, which is a very popular and interesting problem to work on. There are many approaches out there. Briefly, what this approach does is takes as input gene by cell matrix and feeds that into a network inference algorithm, and then out comes a network connecting regulators to target genes. And there are several methods out there and several good reviews covering these different methods, as well as several benchmarking studies that have tried to compare these different network inference methods for both simulated and real data, including one from our group. The challenge, however, in terms of using these approaches for inferring networks for a cell lineage is that the majority of the methods have treated this as an independent learning problem. What that means is that they look at individual cell types at a time and infer GRNs for each of these individual cell types, but not really leveraging the information that these cell types actually related by this kind of a tree structure. So this way of solving this problem is called single task learning, and this can be difficult, especially for looking at network dynamics, because we know that these networks can be noisy and made in for very, very different networks. And the other issue is that the majority of the methods have used primarily single-cell RNA-seq data sets, and very limited amount of integration has happened where, for attack-seq data to infer these gene regulatory networks. So to address this problem, we developed an approach called single-cell multitask network inference, or scMTNI for short. This approach takes as input single-cell RNA-seq data sets for individual cell clusters and also a lineage tree. And also, if we have single-cell attack-seq data matched for each of these different cell clusters, then we can provide that as input. And then the scMTNI framework takes these different inputs and then outputs a gene regulatory network for each of the different cell types, and this does this in a way such that it leverages this information of how the cell types are actually related. So we are assuming that we have a way to actually get these matched cell clusters for RNA and attack, and there are different approaches that are out there to address this problem. And on the lineage prior side of things, which is how we use the lineage structure to do the joint learning, the way we do this is using a probabilistic prior that provides a probability of an edge presence for each of the different cell types. And then we can tune these parameters to control the extent to which the networks should change as we are going down a particular lineage structure. And we can use both, I should say that the scMTNI approach is meant for single-cell attack-seq data, but it can be used for bulk attack-seq data as well as a motif-based prior network. And it can be used to infer these cell type-specific networks by leveraging the cell type-specific gene expression data. And so in that case, the prior won't be cell type-specific. So we used our scMTNI algorithm on simulated data along with several other algorithms, and we have looked at both methods for multitask learning, which basically leverage the information that we have multiple cell types that might be related and do some kind of a joint learning, as well as methods that look at individual cell types and inferred networks individually. And what you're seeing here are F scores, which higher the bar, the better. And we can see that the scMTNI method is doing very well. And another method that among the single task methods is the scenic method, which also does very well. Now moving on to real data, we applied scMTNI to two data sets. One was from cellular reprogramming in mouse, and this has the attack-seq data, which is novel to our study, and published a single-cell RNA-seq data, and the hematopoiesis differentiation system, for which we have published RNA-seq and attack-seq data. So here's the workflow, starting with input single-cell RNA-seq and attack-seq data sets. We use Liger to do integration to define cell clusters with matched single-cell RNA-seq and attack-seq profiles. And then once we have the cell clusters, we apply a minimum spanning tree approach to infer the lineage structure. And then we applied different algorithms, which were different variations of the scMTNI framework, as well as scenic, which, as I showed in the previous slide, was one of the high-performing algorithms. And then we also analyze these different networks in terms of the dynamics to look at which parts of the network actually change on the lineage structure. And the output was prioritized regulators as well as network components that are changing on these different lineages. So I'm going to describe these results using the cellular reprogramming system. This is something that we are studying with our collaborator, Rupa Sridharan. This is a mouse system where we are going from mouse embryonic fibroblasts to the pluripotent state, and we have a total of 6 time points, including the starting MEF cells and the end pluripotent state. And we have single-cell RNA-seq and attack-seq for each of these time points. And these are some of our team members who are working on this problem. So here, what you see are the Liger clusters on the left and the samples on the right on the UMAP. And what we can see initially here is that this particular cluster shows the nice clustering of the mouse embryonic stem cells, which are the pluripotent state, as well as this cluster, which is largely composed of MEF cells. And so this shows that there's a good amount of integration of these data sets when it is possible. And then there's also these other clusters represent a mix of different time points and good integration of different data sets of the RNA and the attack profiles. But it's quite a mix, which is expected given the dynamics of this system, which is quite a heterogeneous system. More clearly, we can look at the individual clusters as well as their accessibility composition, as well as the transcriptomic composition of each of the different clusters. And again, here we see the C4 cluster is the one that is associated with largely the MEF cells, and the C5 cluster is largely composed of the ES cells. And so we took the RNA-seq measurements for each of these different cell clusters, obtained a pseudo-bulk profile, and then learned a minimum spanning tree lineage using the pairwise distance from the expression vectors. And so here's what you see, what we get. So here, the cluster C4 is largely the MEF cells, and then this cluster is largely the ES cells. So this would be the trajectory of cells becoming pluripotent, whereas this is another trajectory that we find where the cells do not reach this pluripotent state. And so we looked at we applied scMTNI along with these other algorithms to these individual clusters in a joint learning framework as well as in an independent learning framework. And so the in-depth method is basically scMTNI, but without any lineage prior. And what we see here are F-scores, again, using a CHIP as a gold standard, Perturb as a gold standard, which are perturbation, upregulation, up expression or down expression of the regulators, as well as the intersection of the Perturb and CHIP data sets. And what we see is that for these two gold standards, the scMTNI method is able to do much better, especially when it is using priors compared to other approaches, which suggests that there is an advantage in terms of using multitask learning as well as incorporating priors. In the CHIP data set, we found that scMTNI was not doing very well. The scenic algorithm was doing much better. And we think this is largely because the types of regulators that scMTNI is able to pull compared to scenic, and we found that there was a low overlap in the gold standard compared to in the scMTNI prior network compared to what we found in the scenic network. So now moving on to the question of how the networks actually change, that is looking at the dynamics. And so we have developed a framework which is really adapting an approach that was previously developed for looking at cell type specific CHIP-seq data sets used to infer TF target networks. And we've adapted that to for single cell networks. And so the overall approach is to use something called a latent Dirichlet allocation. And what we do is we take the individual cell type specific networks and concatenate them or merge them so that we have as many cells, as many rows as the number of transcription factors or regulators times the number of cell types, and as many columns as the number of targets. And we feed that into LDA and we get out these topics, which we then analyze to look at these different network dynamics. So to tell you a little bit more about that, the latent Dirichlet allocation framework is a probabilistic method to cluster different documents. And here it's assumed that each document is defined by a composition of k topics. So k is a parameter to the algorithm. And this is captured by this theta parameter. And then each topic in itself has a different distribution of different words, and that is captured by the beta parameter. And the algorithm estimates the theta and the beta parameters from the data. In the case of gene regulatory networks, our documents are actually treated as each of the transcription factors. So each transcription factor is treated as a document, and each target is treated as, or a gene is treated as a word. And what we are assuming is each of the topics becomes groups of genes are corresponding to different pathways. And when we pass this through the LDA algorithm, what we get out are these estimations of theta and the beta parameters. From the beta parameters, we can get out the specific genes that correspond to each of the different pathways. So in this case, X1, X2 correspond to pathways pathway P1, and the other ones correspond to P2. And on the regulator side, we have a different a row for this theta parameter, which tells us the probability with which this particular transcription factor was generated by this particular topic. And we can compare these individual rows for each of the cell types, and then we can get things like this is a rewired regulator because this particular regulator changed its association to pathway from this cell type to this other cell type, whereas in this case, it largely remained the same. So in this way, we can try to analyze this data and look for regulators that are rewired, and then the pathways or the topics can tell us what are the different subnetworks that are associated with each of these potential different networks. We looked at the first question of rewired regulators, and here we compared the cell clusters C4 and C5, which, if you might recall, C4 corresponds to the MEF cluster and C5 corresponds to the ESC cluster. And we compared, so these each of the rows correspond to the probability of a particular regulator to belong to a particular topic. And we computed the distance between each of these different vectors for each pair of cell types, in this case C4 and C5. And we ranked the regulators based on their difference. And this is very similar to what was used in the topic net paper. And using this, we can actually find what are most rewired regulators. And what I'm showing you here are the top 50 rewired regulators. And this includes several known ones, such as SOX2 and KLF4, which are known to be important in the ESC state, as well as Thi1, which is a MEF-specific gene, and then WT1, which was a new regulator that we identified, and we'll talk about this in subsequent slides. So beyond the pairwise analysis, what we would really like to be able to do is look at how the gene regulatory networks change as we go from this MEF state to this pluripotent state, as well as what are the gene regulatory networks that actually differ between cells that go on this path versus cells that go on this path. So the topics actually give us this information, and so what this is showing you are the parts of the gene regulatory network for each of these different cell types that correspond to one of the topics, topic 3, and these little blue dots correspond to genes, and the labeled dots are the different transcription factors. And what we see is the emergence of certain transcription factors such as ESRRB in the cell cluster C1 and C5, which are the ones that we associated with cells becoming pluripotent. And so this was nice to see because we know that ESRRB is a key regulator of the embryonic stem cell state. Beyond that, we can also compare these networks and see what regulators are likely present in this other lineage. And what we see is this regulator that we also saw in our pairwise analysis, the WT1. All of this is kind of summarized over here, the different regulators along with the different cell clusters. The size of the circles corresponds to the number of targets that are present in that regulator in a particular topic. So the WT1 regulator is interesting in the sense that it has known roles in as a tumor suppressor as well as for different developmental transcriptional programs. And one could hypothesize that this WT1 in some way interferes with cells to become pluripotent. And we could do an experiment, which is something that we are planning to do, where we knock down this regulator and see whether that actually affects the reprogramming efficiency. So now I just wanted to summarize the second part, which is to get to the question of defining the cell type specific gene regulatory networks. And I talked about a method, scMTNI, where we leverage the lineage tree structure as well as a single cell tax-seq data to infer cell-type specific gene regulator networks and also look at their dynamics. And we looked at how these networks behave in cellular reprogramming system as well as hematopoietic differentiation. And we were able to identify several key regulators, both known as well as novel ones that might be interesting in terms of defining what might be the different regulatory programs that are governing the different cell fates. And finally, I want to talk briefly about this question of how accurate are the GRNs. So this is really a validation question. But I'm going to do a quick detour and tell you about some of our efforts for mapping gene regulatory networks in bulk transcriptomic data, where we and others are developing methods to use bulk gene expression data together with other auxiliary types of data sets to infer gene regulatory networks. And something that we've done and others have done is to actually use these auxiliary data sets to infer hidden activity levels or transcription factor activity levels, and then feed that into network inference. And our method is called MEF-PTFA. And what that enables us to do is incorporate regulators that may be under post-transcriptional or post-translational regulation that may not be changing that much at the expression level, but still may be playing an important role in this gene regulatory network. So if you want to hear more about the MEF-PTFA framework and its application, I encourage you to look at Spencer Halberg's talk in NetBioCosi, and also check out this poster where we talk about the MEF-PTFA algorithm and its application to another system. But we actually applied this in a mammalian setting for mouse embryonic stem cells and also human embryonic stem cells by leveraging gene expression data that are publicly available. And this was before using actually single cell data, before that was actually available. And so we used that to infer a gene regulatory network using MEF-PTFA and then prioritize 60 regulators. And then we asked the question, are the regulators actually important for embryonic stem cell state? And also the networks that we predict, how accurate are they? And together with Rupa, who's our key collaborator here, we did some validation experiments. And what I'm showing you here is one of the regulators. We had a total of 8 regulators. This is ESRRB again. And here, what we found was that based on changes in gene expression measured using RT-PCR, we were able to validate several of our computationally predicted interactions. But this was all really low throughput, as in we validated only 8 regulators. What we would really like to be able to do is do this in a high throughput manner. And this is becoming increasingly realistic thanks to recent advances in technologies such as Perturb-seq, which enable us to perturb individual cells with individual regulators, perturb regulators in individual cells, and then measure on a genome-wide scale what the transcriptional program looks like. And so we decided to use this, and what I'm going to tell you about is some early work in trying to use the Perturb-seq assay to validate some network predictions. So as input, we took again the same prioritized regulators, and we made a guide library. And then we are treating day 0 as the transduced time point at which we transduce cells, and then we collected RNA-seq data at 2 time points, day 3 and day 11. And from that data, we identified what regulators are associated with what genes using some analysis that I'm going to tell you about. And then once we have this, we then compared this to what we inferred using our MEF-PTFA algorithm. So initially, some stats. What you're seeing here, this histogram shows the distribution of the number of cells for each regulator. So we have we ended up using about 52 regulators, and what we see here is the number of cells that each that a regulator is associated with based on the detection of the guide RNA for that particular regulator. And there are on average about 500 cells, which is a pretty good number in terms of doing downstream analysis. And then on the right is a histogram that shows the number of guides, number of perturbations that a particular cell sees. And here we want this number to be low because you don't want to perturb a cell with too many regulator perturbations, as that can get confusing. And what we see is that the majority of our cells are actually seeing one perturbation, and there are a small number of them that actually see more. So what you see here are all of our data together visualized in a force atlas plot. And you see 4 types of cells here represented by these different colors. So each color represents either whether they're coming from the day 3 time point or the day 11, whether they're perturbed or unperturbed. And by perturbed, we mean that they saw a guide RNA versus not. And what we see is that all of the perturbed cells tend to cluster in this part of the visualization compared to the unperturbed clusters. And what we could infer from that is really that the cells that are seeing a guide RNA are likely exhibiting a change in the transcriptional program that is different from the unperturbed cells. And we further cluster the data using NMF, and we've into 15 clusters, and you can see the composition of the different perturbed cells and the unperturbed cells across these different cell clusters. And we find that there are 2 main clusters, 1 and 2, which have the majority of the perturbed cells, and they're also enriched for these different guide cells that have the guide RNA. And so this is just a simplified view of the data, where I've just colored the cells based on whether they were perturbed or not. And we looked at the expression level of key marker genes for ESC state. And what you can see is that in the yellow cells, largely the several ESC genes tend to exhibit a lower expression compared to where the unperturbed cells land. And this is clear for NANOG and ESRRB. And what this suggests is that the perturbed cells exhibit a transcriptionally different signature, and they are changing their overall ESC state. And this can be useful to look at how the underlying gene regulatory networks might have changed. So now that we have our perturbed versus unperturbed cells, what we did was we tried to define regulatory connections between transcription factors and targets by doing simple things like looking for 2-fold change between the perturbed versus control cells in the same cluster, or 4-fold change, as well as significant change in expression using a cutoff of 0.05. And we compared the Perturb-seq networks to known interaction networks that are available from the ESCAPE database as well as from literature. And what you see here is an F-score that shows the extent of agreement between what we are obtaining from Perturb-seq versus what already is there in these different databases. And these numbers are actually much higher than what we see when we compare experimentally inferred networks with computation inferred networks. And then here is the comparison of Merlin networks at 2 confidences, 60% confidence and 80% confidence. And in blue is the comparison of the Merlin network and ESCAPE. And we see that the F-score for the ESCAPE network is higher compared to the Perturb-seq network. However, the Perturb-seq network is actually much bigger, and so we can say much more about our Merlin networks, and we can see that for both of these networks, a large proportion of our predicted interactions are actually exhibiting a 2-fold change or higher in our Perturb-seq networks. And furthermore, we can say much more about the transcription factors. So there are more transcription factors for which we are able to observe this change in expression compared to what we could say in the ESCAPE database. And then finally, here is the network that we inferred. This is the Merlin 80% confidence network. And here the different edges are colored based on whether they were in the Perturb-seq 2-fold network, in the ESCAPE network, or both. And we can see that many of these edges actually are green, which suggests that they are supported by Perturb-seq. And the ESCAPE network is largely concentrated around this area. And furthermore, we can see that this particular network has several regulators whose targets you're able to validate, such as POW5F1 and SOX2, and that which we know are known important regulators for the embryonic stem cell state. So with that, I want to summarize what I talked to you about today. So this problem of defining gene regulatory networks for cell fate specification requires us to accurately look at what are the cell types, how are they related, and also identify the underlying gene regulatory networks and their dynamics. I talked about two methods, TMF to look at heterogeneously related data sets, to integrate and such data sets and define cell clusters, as well as the scMTNI method, which can be used to infer gene regulatory networks from single-cell RNA-seq and ATAC-seq data, and also some ways to try to look at their dynamics. Going forward, I think one of the things that would be really important is to carry out more and more systematic high-throughput validation by leveraging these high-throughput assays, and do this in an iterative framework so that we can continuously improve the models that we are actually creating. And something that I didn't quite talk about is this question of lineage inference. And this is something that we assumed as a given, and this is also an important problem, especially in the context of trying to integrate RNA-seq and ATAC-seq to do a good job of inferring the lineage structure. So that would be another direction of work that would be very of high interest to us. I wanted to end by acknowledging my key collaborators, both at UW-Madison as well as outside of UW-Madison, and also my group who have been doing all the work. And if you're interested to actually know more about some of our work in gene regulatory networks, especially for omic data sets, I encourage you to check out these different posters, thank the different funding agencies, and thank you for your attention.